So without further ado, I want to call our next speaker, Nacho Bravo, and he's going to talk about papillomavirus phylogenomics reveals different modes of evolution for the different papillomavirus genes. Nacho.
Well, thank you very much, Robert. And it is indeed a pleasure to be here, and it's indeed to have a pleasure to have a session on papillomavirus phylogenomics. This is my disclosure slide. I don't have any conflict of interest to declare. And the title of the talk I would like to present today deals with papillomavirus phylogenomics and the evolution of the different papillomavirus genes. Which is the objective of this global research, which more or less is try to disentangle which the evolutionary histories of papillomavirus genomes and the evolutionary histories of the papillomavirus genes within the genomes are, and which the connections are, what we can learn from by studying these stories about the relationships between papillomaviruses and us, papillomaviruses and the hosts that they infect. And this from the deep belief that the better our understanding of papillomavirus evolution is, the better our research on papillomaviruses will be. And this for all our research, either basic or clinical or epidemiological. This is a slide that I will be showing very shortly, simply to show you which the current dogmas in papillomavirus research are. For most of them, we do not have an evidence that they are true. There will be a workshop on animal papillomaviruses later, and then we will probably address most of these questions. But then how can we address papillomavirus phylogenomics? What is papillomavirus phylogenomics? We try to understand which the evolutionary histories of papillomaviruses genes and genomes have been by comparing and analyzing papillomavirus gene and genome sequences. The methods that we use for this are methods that you probably may be acquainted with all the research that you do, essentially maximum likelihood and Bayesian methods, with the main difference here that our data are the sequences of the genes and genomes that we are studying. And when we analyze the papillomavirus genomes, this is a phylogenetic tree showing the relationships among the papillomaviruses genome that we know now. As you can see, there are the different papillomavirus genera labeled in the outside of the circumference. And what we can see is that there are 4 main clades of papillomaviruses that I have labeled with different colors. As you can see there, the red clade is a very large one where HPV 16, 18, 6, and 11 and the like that you know very well live. And then there are close relatives of these human papillomaviruses infecting or causing mucosal lesions in whales and dolphins, in pigs, and polar bears. Then there are the other large clade with the green papillomaviruses, which are the beta and the gamma papillomaviruses, and there are also many other papillomaviruses infecting bovine, infecting rodents. And there are 2 small clades, one blue up there and one orange down there. The blue up there, you may know it, some of the members, like bovine papillomavirus 1, that causes febrile papillomas in cows, but also is able to infect horses and zebras and giraffes and buffaloes. And down there, this orange clade that also contains some human papillomaviruses that you may know, like HPV1, HPV63, HPV41, and also other papillomaviruses infecting big cats, panthers, lions, and also some dogs and some other carnivores. So, what I want to say here is that if you look at the small figures, I don't know whether you will be able to see them, is you will see that there are human papillomaviruses in 3 of these 4 main clades. If all papillomaviruses had a single origin, then all papillomaviruses would cluster together in these 3. And we don't see this. This means papillomaviruses, human papillomaviruses, are polyphyletic. They have multiple origins. And this we can see here, for instance, saying that if papillomaviruses had co-evolved with their hosts, then we should be able to plot the 2 stories of the 2 of them, of the hosts on the one side and of the papillomaviruses on the other, and to link them by lines without any single crossing. And then what you see here is there are multiple line crossings when we try to connect the stories of the papillomaviruses and of the hosts they infect. This means that we can bring this into numbers and into a statistical support, but the main message is that there is no support for the hypothesis of a strict co-evolution between papillomaviruses and the host they infect. And this is not trivial, because most of the information that we have about the speed of how papillomaviruses evolve is based on the hypothesis of coevolution. And so, coming back to this slide, I want to emphasize again, papillomaviruses infecting humans are not monophyletic. There is no a single ancestor for all human papillomaviruses. This means that when we start a sentence saying the human papillomavirus causes cancer, the human papillomavirus is whatever, the human papillomavirus has whatever, that sentence is very likely to be false, because there is no human papillomavirus. The human papillomavirus does not exist. There is no an entity that we can pinpoint and say this is the human papillomavirus. So rather, what we should speak about are papillomaviruses, and papillomaviruses infecting humans. And this is not simply a rhetorical question. What I want to say is that if we want to study papillomaviruses that cause lesions in humans and that cause mucosal lesions in humans, most of them being down there in the red clade, then we will be better off by studying animal papillomaviruses that cause similar lesions in other animals, like other on the, up there on the red clade again, infecting and causing genital lesions in whales or in dolphins, or lesions in the tongue of polar bear, or lesions in the pigs, rather than trying to compare and to understand something of HPV 5, say, an example, which is will belong to the green clade, by studying HPV 16, which is in the red clade. This means they may have something in common. Yes, they may have something in common, and there must have been convergent evolution because they are infecting the same host. But most of the things that they will have in common, they will not have arisen by common descent. One of the problems of showing phylogenetic trees is that everything seems as to be very neat. Everything is split. Say a split is very nicely shown, saying here I have a common descent and a branch of a tree, and then it goes to these papillomaviruses here, and then to other these papillomaviruses here. But the truth is much more complex than that, and this, what you can see here, is a phylogenetic network that has been constructed with the same information that I have been showing you before in the phylogenetic tree. And as you can see, the center of the network, this netted region there in the center, is impossible to disentangle. Which we mean is that we can try to track back how the evolutionary histories of papillomaviruses, genes and genomes have been, but we cannot go very deep there in the center. And this is true not only for all papillomaviruses that I am showing you here, but if we go deeper, for instance, to alpha papillomaviruses, which are papillomaviruses that most of you are interested in, is that this is the phylogenetic network for the alpha papillomaviruses. As you can see, there are these three large groups, one with HPV 16 and the like, one with HPV 6, 11 and the like, and one with HPV 2, 3, 10, 27 and the like. And HPV 54 there in between that I do not know what it does. It's a strange virus. It would be very nice if some of you could take some time to have a look at it. But what I want to say here is that there is a connection between evolution, between the genotypic diversity and the phenotypic manifestations of the infections of these viruses in their hosts, as you can very nicely see here, that viruses causing similar lesions or with similar outcome of the infections tend to cluster together. But what I want to stress here is that there is not a neat history. The netted region there in the center speaks of a recombination event that occurs somewhere between 25 and 30 millions of years ago, and that, is related essentially with E6 and E7, and probably also with E5, and I will come to this point later. But again, what I want to say is that disentangle the evolutionary histories is not easy, and maybe we may not be able to do it. And then when we try to study papillomavirus phylogenomics, this means how the genomes have evolved and how the genes that are within the genomes have evolved. One important thing is that the genomes are more than the mere sum up of the genes that live within. And when we study now, for instance, the evolution of the genes within the papillomavirus genomes, you will see there in the x-axis is nucleotide variation, in the y-axis is amino acid variation. The level at 1 means how the average papillomavirus genome evolves. So this means then that E6 and E7, they evolve about 2 times faster than the average of the papillomavirus genome, while E1 and L1 are much more conserved. And this refers for all papillomaviruses, but the thing is that when we go to shallower levels of the tree, the image is not necessarily the same, and this is the same plot for the variants of HPV 16. And as you can see here, E7, that before was evolving 2 times faster than the average, here most of the variation accumulates at the nucleotide level and none of them at the amino acid level. And here most of the variation of the differences between the variants of HPV 16, they accumulate in the E5 genes. And when we analyze multiple of these markers and try to compare how the genes within the genomes of the papillomaviruses have evolved, what we get is that there are 3 main blocks in the papillomavirus genome. And these 3 blocks, they contain genes that have similar histories, which are the oncogenes E6 and E7, on the one hand, the E1 and 2 genes involved in DNA replication, and the L1 and L2 genes. This means then that the papillomavirus genome as a whole, then it contains blocks that evolve and that have different histories. So one of the ideas and of the objectives is try to disentangle or to have an idea on how the evolution of papillomaviruses may have occurred. To my understanding, the best resume of this is that somewhere 300 millions of years ago, when amniotes, so all animals these that lay eggs like us, appeared, there was already ancestral papillomaviruses that infected these ancestral amniotes. Then, during the development of mammals, one of the particular things of mammals is our skin. We developed hair, we developed sweat glands, and in my eyes, these created different niches that papillomaviruses colonized and radiated and colonized, and these generated a number of papillomavirus lineages that corresponds to the large 4 main clades that I have been showing you before in red, blue, and orange, and green. And then later on, somewhere around 100 millions of years ago, mammals, us, we radiated ourselves. And then papillomaviruses followed us somehow, so that the diversity of the papillomaviruses that we see now, it corresponds to the colonization of these new niches in the skin, plus the radiation of us as the hosts. And then later on, in particular branches of the PV tree, there have been particular events, like for instance, the jumping in of the E5 genes into some of the alpha papillomaviruses, of the jumping in of the E5 genes in the delta papillomaviruses, BPV1 and BPV2. And then, in my eyes, these events have occurred separately and do not have are not a single event, meaning that the E5 proteins in the alpha papillomaviruses do not have a common origin with the E5 proteins in the delta papillomaviruses. Maybe we should not study them together. And then, again, going back to the main idea that the better our understanding of the evolution of papillomaviruses is, the better our research at all basic clinical epidemiological levels will be. These are the conclusions mainly of the whole talk of the existence of different genes within the genome, these blocks within the genomes of the papillomaviruses, and the existence on how the diversity of papillomaviruses has occurred by radiation and by partially following their hosts, and that one claim for broadening the diversity of papillomaviruses that is known. And this is what all I wanted to say until this moment, and then I guess, Robert, that we will address the questions at the end? Okay, thank you very much.
You can either sit here or you can go down and come back. Okay. So, thank you very much. I would say that I think in general, he gives us a lot of important information, and that the coevolution aspect of papillomaviruses really has occurred subsequent to the adaptation to ecological niches, such as the cervical vaginal area. And then we see a relatively strong signal for coevolution. But taken as a whole, I would agree completely that it's a much more complex story.